So blood. We've got a lot to cover. Obviously it's Monday. Shake off everything. It's funny. I look out and not that I want this to be on the lecture. I see it in your guys' faces, and again, I can't thank you guys enough. I can't. I mean, I really can't. You guys were there for me, and it made a huge difference. So I appreciate that, and I will keep doing what I can do to help you guys out, no matter what. So blood. Now, all the things that I talked about on Friday with blood, those still stand. But what we're going to do now is we're going to start tearing it apart. Now, you guys should study this in two ways. Use their technical names, but for right now, all I'm going to do is... Oh, seriously? Okay, so I'm going to use their abbreviations, but I still want you guys to use the technical names. You should split it up like this. Okay, but you should be using their technical names. So erythrocytes or leukocytes. Okay? Erythrocytes or leukocytes. Erythrocytes means red blood cells. Leukocytes means white blood cells. Okay, these abbreviation, white blood cell, or white blood cell, red blood cell. They also have another abbreviation when you're in healthcare. Red blood count, white blood count. Okay, so bless you. So just keep that in mind when now it means cell, but when you move into healthcare, it actually means count. And you'll see it if you become a phlebotomist or you've done any phlebotomy, you'll see it. You'll totally see the counts on there. And I have a student in the Saturday class who is a phlaebotomist, and it's fantastic because she literally aha moments continuously in the classroom because she's linking everything together, and that is very special. When you're doing a job and then you get to link everything together. Okay, so an introduction to blood and the cardiovascular system. Well, the cardiovascular system consists of the heart. It's a pump. Well, I hope so, because guess what? It never, ever stops. In fact, I have a little thing for you guys to do here and see how it works. Did I do that with you guys already? The pump thing? No? Okay, good. So a conducting system, which is the blood vessels, and like any conducting system, you can what? You can let more in, or you can reduce the amount that's there, conducting. And then a fluid medium, which is blood. Blood is a fluid medium. It's specialized fluid of connective tissue. We know that from 241. All right, it does have fibers. It does have extracellular matrix. It does have cells in it, so it does make it. Now, when do the fibers come out? It's not a trick question. Blood clotting, yeah, fantastic. Okay, and then contains cells suspended in a fluid matrix. Now, blood itself transports materials to and from the cells, oxygen, carbon dioxide, nutrients, hormones, immune system components, and, of all things, waste. Now, CO2 is considered a waste, even though it's a gas. Okay? But we do get rid of waste products. Now, nutrients, hormones, we just got out of hormones. We'll get into nutrients when we go to the digestive tract, and then immune system. Well, that's part of this, and that immune system is going to be huge. Okay, and it's pretty fun too. I don't know if I can get the school to set it up. If they can, we'll do it. We'll do an epidemic lab. Okay, basically we'll infect one of you, and then you will come in contact with other people in the class, and we'll see how many people get infected. So if I can set it up, I will, and we'll do that here at the school. It doesn't take that long to set up, so we'll see what happens. Important functions of blood. Well, transportation of dissolved substances. Regulation of pH and ions. So, what's the pH of blood? There we go. 7.35 to 7.45. Restriction of fluid loss. There's our clotting. Okay. Defense against toxins and pathogens. All right. And then stabilization of body temperature. Heat regulation through sweating. Okay. Circulation of blood from sweating. Now, everybody thinks of heat regulation when heat, if the word heat's there. But if you're really cold, guess where all your blood goes? Into your core. Okay, it's like, you know what, I don't care if you lose a nose or a finger or anything like that. This is important. And it will shunt all the blood to the core. So whole blood, well, plasma, what percentage of plasma is water? Oh, no, plasma is 55, 90, there you go, is water. So, Mohammed jumped the gun, but that's good. We'll elaborate on what he said. He said 55. Well, of whole blood, 55% of that's plasma. But of that plasma, it is 90% water. Okay? Fluid consisting of water. So just put a gigantic 90 behind that. 90% dissolved plasma proteins. Okay? Well, we're going to talk about proteins, so you might as well, right behind that word, write the word colloid. Colloid. Okay, because that's going to come up. Right? We have specific proteins that we'll focus on. The primary proteins, albumin. Albumin. Actually, let's put that up there too. Why not? Albumin. N. Just an N. N. Albumin. Okay, and primary protein. We'll be talking about that one and why protein is so important. Then it says other solutes. Well, yeah, we'll touch on them, but nothing major, nothing to get excited about. And then formed elements. Well, cells and solids. Well, if we're talking about blood, yeah, red blood cells, white blood cells, platelets. All these wonderful things found in blood. So, boom. Now, 92%. Whoa. I guess I skimped on 2%. All right, so here we go. Plasma proteins, about 7%. Other solutes, 1%. Water, 90%. 92, 90, right in there. Okay? Every book's going to be different by 1 or 2%. It's not a big deal. But just remember, it's a large amount. Then we drop down, less than 1%. Less than 1% is platelets. Less than 1% is white blood cells. And then 99% of this 45 is red blood cells. So in the formed elements, 45% red blood cells, and then in that less than 1% for platelets and white blood cells. Now, notice here it has a percentage. It actually says 37 to 54. 37 to about 45 is the percentage for women. 45 to about 54 is a percentage for men. Does anybody know what it's called when we separate out the red blood cells and do a percentage? Anybody? It's called a hematocrit. A hematocrit. Okay, and we can do that in 5 minutes. So literally, if you're in a motor vehicle collision or you fell off of something or anything like that, the first thing they're going to do is they're going to do a hematocrit, because that tells us if you're losing blood. Now, obviously, if you have a huge gaping wound and blood squirting out of it, I don't think we have to do a hematocrit. I think we have to stop the bleeding. But if we can't see any damage, and you said you were in some type of blunt trauma damage, we're going to do a hematocrit, because you could be bleeding internally, and we need to know that. Right? The lowest I've ever seen in that is about a 21%. That's the lowest, and it was because of a motor vehicle collision, and what happened was the person struck a telephone pole. Okay, their seatbelt worked, but they were going so fast that they did hit the steering wheel. Okay, and it actually the steering wheel hit them because it collapsed in on them, and it punctured their heart with one of their ribs. So they were literally pumping blood into spaces, and they were at 21 when they got to the hospital. So they saved them. They actually went in there and plugged the hole, sutured it back up, and straight to surgery. But yeah, it's pretty crazy when you see that stuff. Had we waited, probably dropped down to 10 and they would have died, because all the blood would have been in the spaces. Okay? So 3 types of formed elements: red blood cells, white blood cells, and platelets. There you go. So red, white, and we've got our colloid, albumin over there, but then platelets, and it's all about clotting when it comes to platelets. Now, there are studies out saying that platelets have a lot more to do than with just clotting. They have some to do with the immune response, okay? They have some to do with health. They chemical messengers to tell your body something's going on. So there's a lot more going on with platelets than just clotting. But for most of the time, when we hear platelets, yeah, we're thinking clotting. Okay? Hematopoiesis, the process of producing formed elements by myeloid and lymphoid stem cells. Myeloid and lymphoid stem cells. Myeloid and that lymphoid stem cell stuff there going on. Myeloid, lymphoid. Myeloid, lymphoid. Okay, just saying. All right. Fractionation, the first time I've heard this, but that's okay. We'll use the term in this setting. It's this process of separating whole blood from clinical for clinical analysis into plasma and formed elements. Bless you. It's like pizza, but for sneezing. Okay, so basically what we're talking about here is spinning your blood down, separating your blood, centrifuging your blood, separating it out. And as you spin it out, guess what happens? Plasma goes one way, blood goes the other way. Okay? It says 3 general characteristics of blood. Well, there's our temperature, 38 degrees Celsius, 100.4 is the normal temperature. High viscosity. Okay, it's pretty thick. So that's why we need that water in there. All right, and then slightly alkaline. And there's our numbers. Those don't change, that's good. I'm glad they didn't do like some 7.36. It is 7.35 to 7.45, slightly basic. Slightly basic. Any questions so far? This is just kind of an overview of what it is. I mean, we're going to break it down for each little part, but yeah, just a little bit of an overview. Questions? All right. Characteristics of blood, well, blood volume in liters, 7%. 7% of the body's weight. Wow. That's pretty amazing. 7% of the body's weight is blood. Okay, in adult males, 5 to 6 liters. In adult females, 4 to 5. Those numbers you do need to know. Okay, when we talk about circulating blood in the body, you need to know how much is circulating at a certain amount of time. So we're going to have math problems. I know you all love math problems. They're pretty simple. They're not crazy. They're not out of control. They're pretty simple. It's a little bit of addition and some multiplication, and you're solid. No fractions. I'm not into fractions. I hate fractions. No using letters. Whoever thought of putting letters in math should be banished to an island somewhere. That's ridiculous. Really? Find X? It's at the end of the alphabet. That's all I need to know. Okay? Find X. Oh my goodness gracious me. Whoever thought of that? Silliness. Silliness. Put a number in there. It's called mathematics for a reason. X. It's somewhere down at the end of the alphabet, and I still have to do this. LMNOP to get the right one. All right. Composition of plasma itself. Well, it makes up about 50 to 60% of blood volume. More than 90% of plasma is water. So there's our 90%. Okay, notice I said more, but there's the 90% we needed to see. All right, so plasma, 90% water. Extracellular fluid, interstitial fluid, and plasma. Materials, plasma, and interstitial fluid exchange across capillary walls. This is pretty cool. Okay? Waters, ions, and small solutes, not proteins. I mean, some protein, but not big proteins. Like albumin stays in there. Small proteins can diffuse across. Yeah. In fact, our capillaries, and what they're talking about here across the capillary wall, our capillaries are so small that guess what all of the red blood cells do? Single file. They all line up in single file to go through. That's some craziness. Our capillaries are so small that they line up in single file and process through. Now what they're talking about is in the capillaries. Hello, capillaries. Yeah, okay, you guys, let's do a realism. This is it right here. Okay, so everybody, just drop what you're doing, put your pants down, everything like that. You're not going to hurt yourself. All right? And where are your partners? Rawr. I know. Okay, so 2, 2, 2, 2, 2, 2, 2. Oh, I might make you walk. Or I might make you walk. I don't know. Who likes to walk more? You'll come up front? Awesome. Come on up front. Turn to your partner. Okay? What I want you to do is I want you to stick your finger out in front of your partner, and I want you guys to do this. This is something you have to know anyways. No E.T. or anything like that. Come on. They're all silliness. And then all I want you to do is you take your partner's fingernail bed and depress it, and then let up and watch what happens. Now, go ahead and do it. You're going to... Oh, jeez. If they have fingernail polish, let me get the remover. Okay? So squeeze it. What is happening? What is happening when you depress it and let go? Blood circulates back into those areas. So this is how fragile, okay? This is how fragile your capillaries are. Just the depression of your finger is enough to squeeze out the blood that's in there, okay? Just the depression is enough to squeeze it out, and then when you let up, the blood rushes back in. Notice I said rushes back in because in some patients it does not. Okay, they have perfusion problems. Okay, especially distal perfusion. We'll do toes and fingers. So that is called capillary refill, and it's a standard test on every single ambulance. Even in a hospital, when I'm walking through to work with patients in the emergency room, I will walk right up, especially if they're youngsters, and grab their toe and hee. Really? Am I just doing that to make them laugh? No. I'm doing it because I need to make sure that they're perfusing properly. I need to make sure that the oxygen we're giving them is making it all the way down to the areas, distal areas of their body. Thank you, sir. So it's extremely important, and it also shows you how delicate they are. Just the depression of your fingernail causes the blood to actually move out of the capillaries, and when you release it, it moves back in. Okay? So, and that's what they're talking about. Now, interestingly enough, red blood cells can't move out of the capillary unless you damage them. So, now we know what bruises are. It's damage to the capillary, and the red blood cells move out of the capillary into the extracellular space around it, and that's where a bruise comes from. And because it's a process, macrophages have to go in and eat up all the red blood cells. They have to clean up all the debris. So it takes what? It could take up to 2 to 3 weeks to get rid of a bruise. And that's the process. So that's what's happening right here. Plasma proteins. Oh, I guess I misspelled albumin. It's an I. I like the E better, but it's an I. So let's put our I in there. Albumin. And that's the one you guys need to focus on. Okay? That's the one you need to focus on. Plasma proteins, albumin. Now, fibrogen we'll talk about. It'll be a primary protein. What's in that word? Fiber. So, guess what? It's part of clotting. Okay? We need those clotting factors, or not factors, but we need the fibrogen to help clotting during the clotting factors. And then globulins, we'll express that a little bit later, but albumin, that's your pal. Okay, and primarily what he's going to do is he's going to stay in the capillaries, and he's going to help pull fluid back into the capillaries. Okay, because fluid's going to leave, okay, and then he's going to help pull it back. Because we have to have a certain amount of fluid circulating around in our bodies. Okay, so albumin transports substances such as fatty acids, thyroid hormones, and steroid hormones. There's our globulins, antibodies. So that's in the immune system, and that's where we'll get back to those guys. Okay? Immunoglobulins transport small globulins, hormone-binding proteins. Okay? But yeah, antibodies. And we'll talk about those antibodies later on, and there's a whole list of them, and I'll give you a way to memorize them. And then fibrogen, molecules that form clots and produce long, insoluble strands of fibrin. So this is the fibrin mesh that does this, and guess what gets caught in it? Red blood cells. Red blood cells get caught in that, and that's what stops you from bleeding. Okay? So Fibrogen, and we'll talk about that because we'll actually have a section on repair. But interestingly enough, if we go all the way back to tissues in 241, didn't we have a section on repair there too? Yeah, this will be a little bit more elaborate though. Serum, liquid part of blood sample, in which dissolved fibrogen converts to solid fibrin. That's an important statement because that's one of the primary statements for clotting. Okay, so the liquid part of blood sample in which the dissolved fibrogen converts to solid fibrin, all right? Fibers are present during clotting, making this a connective tissue. Wow, I said that in 241. Now we're bringing it up again. So this is awesome for the big picture. This is gigantic because now you guys are taking it in like this. Wow, we've gone full circle. We talked briefly about it because it was a connective tissue. Now we're bringing it back and showing how it works. Oh, recall point characteristics of connective tissue. Really? Why don't I put a flower on there too? Like, hello, Daisy. Yeah, it's a recall point, and that's why I'm recalling it, because it's something you guys actually need to recall. All right? Origins of plasma proteins. Well, more than 90% are made in the liver, antibodies made by plasma cells, and then peptide hormones made by endocrine organs, which we just got out of. So we're going to touch on that a little bit. RBCs make up 99% of the blood's formed elements. Hemoglobin. Heme. Okay, red pigment that gives the whole blood its color, binds and transports oxygen and carbon dioxide. Dioxide, not monoxide. Okay, we're not in a car. Not going to work for you. Dioxide. Okay, hemoglobin. Abundance of RBCs. Well, a red blood cell count, the number of RBCs in 1 microliter of whole blood, male 4.5 to 6.3 million, and female 4.2 to 5.5 million. Now, those numbers can fluctuate greatly in a female, not in a male. And we all already know what I'm talking about. Okay, menstrual cycle does wonders for your red blood cell count. Okay, in fact, that's one of the first questions when we're doing hematocrits, and it comes in, let's say 5 or 10% lower than normal. I'm going to say, are you menstruating? Some people can't take it. Some people can't take it, but you know, actually as you get older, you're like, whatever, yeah, okay. I've enough times in life, yeah. But when you're young, you're still kind of timid by the thing, you know, like, I just got it regular. I don't know what, it scares me sometimes, okay? Yeah, it'll sneak up on you when you're young, okay? But yeah, we have to ask. We have to ask in a clinical setting because that's enough to skew the numbers. It is actually enough to skew the numbers, so we have to ask that in a clinical setting. And mostly in the emergency department, but in other cases we will ask it too. So, all right, so here we go, a hematocrit. There we go, there's our numbers right there. Hematocrit, packed cell volume, okay, percentage of RBCs in centrifuge whole blood. So these are the numbers we're looking for. And again, I wish we could do it in here. It does take about 5 minutes to do it. If we had, do they have phlebotomy here at this school? No, so Central has it, I know, or not, yeah, Central and Votech. Renton has it too. But yeah, you do it in there. You do spin down for phlebotomy. Okay, they do it once too, which is really weird. You think you do it a few more times than that, but I guess they feel phlebotomy is just drawing blood. Okay, it's not hard to draw blood. It really isn't. I think the biggest challenge is actually drawing blood on a child with capillary tubes. That's the biggest challenge. Okay, so this spinning it down takes 5 minutes, and literally we can tell what your count is. Structure of a red blood cell: small, highly specialized discs. Okay, highly specialized discs. So let's turn this guy sideways, and let's just do this. We're going to turn him sideways. Biconcave. Okay? Biconcave. So here's our bi, 1 and 2, and is it caved in? Yeah, it is. Okay, so biconcave, this is what a red blood cell looks like. In fact, if we look at it from the top, it looks like this. Why would it look like that from the top? Yeah, because it's indented right here, so it's super thin. It's actually almost clear in the middle and dark around the outside. Of course, I'll be using red in this case, but yeah, it'd be pink or almost white in the middle and dark red on the outside. So here's your dark red, and there's your thin area where light can come through, and it actually lightens it up. Okay, actually, what does it say? Thin in the middle, thicker on the edges. Okay. 3 important effects of RBCs: shape, high surface to volume ratio. That's because it doesn't have anything in it. It's empty. It's just a container that circulates around for approximately 120 days. Okay? Quickly absorbs and releases oxygen. And now, absorbs is probably not the best word, but for right now, we'll just use that. It's more of an exchange. So when oxygen's asked to release itself to an area, guess what's going to sit down in the chair? CO2. So it's almost like musical chairs if you think about it that way. Oxygen's asked to release itself into the area that needs it, and CO2 says, oh, cool, empty chair. Okay? Discs form sacs. That's not pertinent. I don't care about that. Discs bend and flex, entering small capillaries. So obviously their size is pretty small. They actually have to bend and flex. They can only go through one at a time, and that's what I was talking to you guys earlier. And there we go. That should be a flashback. Tissues, 241, microscope. Trembling because you were new to this whole idea of learning anatomy, physiology. Asking questions like, do I have to learn this? You guys remember those questions? Yeah. That was scary. Now you're all salty. You're like, bring it on. Blood. Got your little walk down and everything. That's right. Whoa, now see, that's more like it. Wow, that looks like your guys' weekly schedule. Right. There's our wonderful little RBC right there. Okay? So I drew it on the board. There he is over there. Biconcave. Lifespan of an RBC. They lack nuclei, mitochondria, ribosomes. Means no repair or anaerobic metabolism. Okay? They eject it. So I'm pretty sure you're all probably wondering, like deep down inside, I was, and I asked the question, okay? And I was told so eloquently by my professor way back when, just wait till I get there. I was like, no, if I have a question, you answer it now, gosh darn it. Okay, and the question was, why do they call it a cell if it does not have a nucleus? It's a good question, isn't it? Because at one point in time, it does. It does have a nucleus. It ejects the nucleus before it becomes a full-fledged red blood cell. Okay? So during its life, as it's developing to become a red blood cell, it ejects its nucleus, gets rid of it, and that's why it's a biconcave, perfect carrier of O2 and CO2. So they kept the name cell because at one point in time it was a cell. It just gave it up so that it could be a more efficient cell. So in this case, it does lack a nuclei, a mitochondria, ribosomes. It lives for about 120 days. Now, no. I had one student say, well, what happens when they all leave? They don't all leave. Okay, they're born at different times. It's like the population, right? We have young and we have old. The old die off, the young come in. It was a cute answer. Most everybody did the same thing you guys did, like. Okay, hemoglobin. Hemoglobin, protein molecule that transports respiratory gases. Normal hemoglobin in an adult, 14 to 18. Normal hemoglobin in an adult female, 12 to 16. Always is going to be a little bit of a difference there. Mainly because of size. All right? Mainly because of size. So, and notice the numbers are a little bit bigger on the high end there. 16, doesn't that fall in between 14 and 18? Yeah, so nothing crazy. Now the structure of hemoglobin, complex structure, 4 globular protein subunits, each with 1 molecule of heme. Okay? Each heme contains 1 iron. So how many are there? 4. Okay? 4 globular protein subunits, each one with a molecule of heme, each heme with 1 iron. So, hemoglobin structure, well, iron ions associate easily with oxygen, oxyhemoglobin, okay, HbO2. This is going to become extremely important when we get into respiratory system. Okay, disassociate easily from oxygen. They disassociate easily from oxygen. Again, this is what we're going to be talking about when we get to the respiratory system. So oxyhemoglobin, that means it's bound. Deoxyhemoglobin means that it is unbound. And there you go. So, hemoglobin molecule, here's a hemoglobin molecule, and then heme, 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 and in the middle of each one is a iron. So, it is no surprise that when you're young, okay, and you're getting ready to cross over into that young woman adulthood time, your mom, maybe your dad, you never know, might have pulled you aside and said, guess what? You're starting to get close to that time when you're going to have a menstrual cycle. You didn't have that talk?
No.
Oh. Thanks, Mom. Thanks, Dad. That's a surprise, like you're in gym class. Ooh, I'm really sweaty. Ah! Yeah, not good, not good. So the idea is, at the same time that they tell you that you have a menstrual cycle, they should also say during that time you should take what? Iron. Because what are you losing? And a large amount of it. Okay? Most of you probably did this. The internet taught me. I got it all from the internet. Google said, and I did exactly what it told me to. Right after I agreed to let it tap into my phone book and call all my friends. Good old Google. Okay, so there we go. There's our numbers. Fetal hemoglobin. Strong form of hemoglobin found in embryos. Takes oxygen from the mother's hemoglobin. Hemoglobin function, well, carries oxygen with low oxygen peripheral capillaries. Hemoglobin releases the oxygen, binds carbon dioxide, carries it to the lungs. Okay, now they're giving us the terms that are more useful in respiratory. So I would put a big R up this at the top of this because you're going to see it again. Okay? A big giant R up there, because literally what they're talking about is the dropping off and picking up of oxygen and CO2. And it is literally like that. So when oxygen gets out of its chair and walks away, guess what sits down? CO2. And we're going to learn about the Bohr and Haldane effect in the respiratory system. We're going to learn how they disassociate. Why? Okay? Because check this out. This is a fully loaded table. Fully loaded table. So are they going to need anything? No. Is this table going to need something? Yeah, it's not fully loaded. Okay? So what happens when you use oxygen, okay? If this table used 1, 2, 3 oxygens and this table didn't use any, I'm not going to circulate over here. I'm going to circulate over here so I can do what here? Drop off oxygen. And that's what they're talking about. Okay? Oxygen's released and the carbon dioxide is picked up. Okay, so that pretty much is our class right there.